Assalamualaikum. My name is Atiq. I am from Department of Zoology, Gazi University, DD. Topic is characterization of lactobacillus species from tiger and leopard species and assessment of their pharmacognostic properties. Introduction. What are probiotics? Probiotics are living microbes, live microbes, in which different type of microbes occur, such as lactobacillus, lactococcus, bifidobacterium, and bulgarius, and so on. As I told you that my topic is on lactobacillus. Lactobacillus are lactic acid bacteria. They produce lactic acid. They are found in our diet. Many types of lactobacillus are found in animal intestine, animal body, and it has so much beneficial effect of animals. Objective of this study was to isolate and characterize lactobacillus species from fecal matter of tiger and leopard to check and compare probiotic tolerance such as pH tolerance, NaCl tolerance, and bile salt tolerance. To investigate in vitro pharmacological applications such as antioxidant property, anti-inflammatory activity, and antimicrobial activity. Material and method. We collected our samples from fecal matter of tiger and leopard of Lahore Zoo, Punjab, Pakistan. After collection, we put it in Pepton water for 24 hours. After that, we put it on media. Media was used MRS with cysteine. Cysteine is good for the growth of lactobacillus, and calcium carbonate is also good to identify the lactobacillus species. As I told you that lactobacillus are lactic acid bacteria. Lactic acid bacteria use calcium carbonate and utilize calcium carbonate, and we can identify by that these are lactobacillus. Bacterial isolation, purification, and identification are done by morphology, biochemical, and some potential tests. Morphological characterization, colony morphology and cell morphology. For identification of lactobacillus, we first of all we have to identify its colony. It has when we collect different microbes, when we see different microbes and we want to collect lactobacillus, then there are so many colonies. We have to identify the colony of lactobacillus. We have to identify cell morphology by different staining mechanisms such as gram staining, spore staining, and acid-fast staining. Gram staining. Gram staining method is done by Colin et al. 2004. In this, we use the method followed by Colin et al. 2004. In which crystal violet, iodine, and alcohol is used. Saffron is used as countercurrent stain. Spore staining. Spore staining method is followed by Harry and Prescott 2002. In this, first of all, malachite green is used as primary stain. After they heat it and decolorize by using water. In the end, we use counter strain saffron to see that spores formation occur or not. In lactobacillus, there is no spore formation occur. So this test will help us in identification of lactobacillus species. Acid-fast stain. This method is followed by Casey and Verma 1998. In this, carbol cysteine is used. After that, heating processes is done. Then acid alcohol is used to decolorize, and as countercurrent stain, we use methylene blue. Biochemical characterization is also used to identify and characterize lactobacillus species. Catalyst test. This test is done by hydrogen peroxide, 3% hydrogen peroxide. If a bubbles formation occurs, then we can say that these are not lactobacillus species because lactobacillus have no ability to produce gas. Carbohydrate fermentation test. This test was done by glucose and mannitol. Potential test, NaCl tolerance followed by Hawk et al. 2010. pH tolerance test done by Hawk et al. 2010, wildfire tolerance is done by using the protocol described by Celia and Maria 2001. Here is some activities, some pharmacognostic activities, antimicrobial activity, antioxidant activity, anti-inflammatory activity. Antimicrobial activity is done by a well diffusion method in agar plate. It is done against three microbes such as E. coli, Streptococcus. Antioxidant assay or antioxidant activity, we done some three essays and three activity to see that the lactobacillus are antioxidant or not, or our strains are antioxidant or not. First one is DPPH, free radical scavenging activity. This method is done followed by Reshma et al. 2014. Hydroxyl radical scavenging activity. This method is done by De Eveler et al. 2004 and superoxide anion radical scavenging activity. This method is estimated by pyrogallol auto-oxidation method, Zhao et al. 2003. At the end, we done anti-inflammatory assay. In this, inhibition of albumin denaturation occur. This method is followed by Reshma et al. 2014 with some modifications. Modifications was done by supernatant and pellets. We did this experiment on pellets and we made in this cell suspension. Here are some results. Colony morphology. Lactobacillus, we isolated two strains. One is lactobacillus from tiger and second is lactobacillus from leopard fecal matter. We collected two strains and both strains have same colony morphology. Colony morphology was same. After sequencing, we know that both species are same, but the sources was different. Here is the result of biochemical characterization. It's gram staining result. In gram staining, the lactobacillus gives purple color because of the crystal violet, because it is gram-positive bacteria, so it gives it gives purple color. Non-spore forming stain. Here we can see that both lactobacillus, the tiger and leopard, both have no spore formation, both have no green color spore, both have vegetative cells which are red in color. Acid fast staining. In acid fast staining, we can see that they give blue color, which means they have no cells like that which can produce tumor, which are non-acid fast. Lactobacillus are acid fast. Lactobacillus are acid fast strains. So by these three staining, we can say that we can identify that our strain may be lactobacillus. Negative catalase test. Catalase test. If catalase test is negative, it means if bubble formation is not occurring with H2O, hydrogen peroxide, it means these are lactobacillus. Lactobacillus, because lactobacillus cannot produce any type of gas with hydrogen peroxide. Next is mannitol utilization and glucose test. In mannitol utilization, you can see that control is in red color. When we put strain in it, when we inoculate it with strains, then it converted mannitol into glucose. It utilized the mannitol and convert it into yellow color. Gas production and glucose test. In this, we use glucose instead of mannitol, and we use Durham tubes. Durham tubes are for identification of gas. You can see that here some bubble formation is occurring, some gas you can see in L and T tubes. There are some gas. It means lactobacillus. This type of lactobacillus, which is collected, which we isolated from fecal matter of leopard and tiger, are gas-producing lactobacillus. Here is potential test result. Potential test, we done three types of potential test: pH concentration, NaCl concentration, biosalt concentration. Lactobacillus are mostly are of different types, and they can alive. They can survive at different pH concentration. You can see that both are giving same concentration of on two, but in this blue color is for strain T, that strain which is isolated from tiger, and this orange color is for that strain which is isolated from lion. So here is the result. You can see that the strain T and the strain L both are giving good production at pH strain 6, pH concentration 6. Next is NaCl. In NaCl, the good production is or on 2. NaCl, we use 2% of NaCl. So lactobacillus give good production, good growth on 2%. Same in bile salt concentration. The bile salt concentration is different because of the different sources. These two lactobacillus, in which strain T lactobacillus, the lactobacillus which are isolated from tiger, give good concentration, good growth on 0.2% bile salt concentration, while red one, the strain of leopard, is giving good growth on 0.4% bile salt concentration. Here is the antimicrobial assay. Both lactobacillus give antimicrobial activity against Streptococcus, E. coli, and Staphylococcus. It is the DPPH radical scavenging activity. Leopard strain gives 33% and tiger strain gives 18% activity against DPPH. Same as in OH ion scavenging activity, the leopard is giving 74% OH ion scavenging activity, while tiger strain is giving 50.6%. And in superoxide radical scavenging activity, both are giving approximately 36.6 and 38.6% activity against superoxide radical scavenging activity. There is not much difference in the activity of both strains. Next is anti-inflammatory assay, in which we done this method to check the inhibition property, to check that protein can be inhibited by protein can be inhibited by denaturation. So in this you can see that the strain which is isolated from leopard is giving high anti-inflammatory activity such as 54%, and tiger strain is giving 17.3%. Conclusion: the strains which are isolated from fecal matter are characterized as lactobacillus species. These are indicated as probiotic tolerant, and these are identified as lactobacillus brevis by gene sequencing after this research, after this study, and both lactobacillus brevis showed pharmacognostic properties but with different percentage. This study enhances the status from probiotic to superbacteria with antibacterial, antioxidant, and anti-inflammatory capabilities. In 3 ways, this will resist us to cope up and preventing a variety of disease. Thank you so much.